You don't want to be seen. So that's the recording on. If you don't want your beautiful face to be seen, please turn your camera off. So today I've got an auspicious pleasure in introducing Malcolm Bennett. Malcolm Campbell. I was speaking to Malcolm Campbell in Guelph earlier on today.
Okay.
My mistake there. The other confusion is, of course, there's two Malcolm Bennetts in Nottingham, isn't there?
There is. I've been called far worse, Derek.
Right. So today we've got Malcolm Bennett from the School of Biosciences at the University of Nottingham. Malcolm's a world leader in root research. I'm not going to go through the bio and seminar synopsis. It's all been circulated and you can see it. I'm sure we will get an engaging talk, and I'll let Malcolm start now. On you go, Malcolm. It's all yours.
Thank you very, very much. Right, let me have a look. I'm going to do the share screen. Jesse was very helpful earlier. Let me run through this. Okay. Can you see that?
Perfect.
I'm going to put you down in the corner there. Okay, thank you so much for the invitation. It's great to be able to join you today and tell you a little bit about a new idea that's come out of our team over the last, I would say, a couple of years. And it stems from this interaction between soil scientists, plant scientists, computer scientists. And you heard a little bit about this last week when you hosted Tony Pridmore. And he's one of the three people, myself, Tony, and Sasha Munoz, soil scientists, who've really kind of driven this area at Nottingham, and this engagement between these different fields, which is so important to understand what's going on in the rhizosphere. So, okay, let's move on. So what I'd like to talk about today is how plant roots sense soil stresses. And, you know, we're aware there are many soil stresses. We tend to focus on things like nutrient stress, but we're obviously acutely aware of soil moisture stresses. So even Scotland experiences soil moisture stresses. So here we have a map of soil moisture over the summer last year, and you can see even Dundee was experiencing drier soils. The west of Scotland, less so, but obviously the east of Scotland, you can clearly say, was joining the rest of Europe in experiencing these issues. And you know, we saw that through, particularly in mainland Europe, through really devastating changes in water distribution, which obviously impacted soil moisture stress. So that's an example of, I guess, climate change having an impact on a soil stress. We're also, soil stresses are created through management practices largely. Soil compaction stress, for example, is a product of poor management. So, for example, using heavy machinery, which compresses and damages, or poor tillage practices. And this obviously can result in up to a quarter of the yield losses. When combined with drought, for example, it can result in up to 75% yield losses. So this is really important. This is a particular problem in Europe. As you can see here, there's a great deal of compaction, stress potential, particularly south of the border. From a Scottish perspective, you can see that, particularly you can see across the UK and also large parts of Europe. So these are just two examples of soil stresses which are obviously impacting plant performance. And what I'd like to do today is talk about how plants sense soil stresses and how they adapt their root growth. And I'm going to focus on one example of this, and this is compaction stress. And I'm going to describe the mechanism for which compaction stress is controlled. Then I'm going to talk more generally about the title of the talk today, which is how this mechanism fits into what we call root radar. And this is a new kind of signaling concept for root soil stress signaling. And I'll describe that in a little bit more detail in the middle of the talk. And finally, I'm going to conclude. You know, I appreciate that the Hutton is is originally was the Scottish Crop Research Institute, and so you were interested in understanding how we can leverage this basic mechanistic understanding and how it can be applied to improve stress resilience in crops. So hopefully I'll satisfy the basic and the applied researchers in the audience today. Okay, so let's talk about compaction. So many of you are aware of this topic area. There's been some great work done at James Hutton in this area. The person I hold up most is Glyn Ben-Goch. He's done some outstanding work in this area, and we've also got, for example, Phil White. I'll allude to their work throughout this talk. So soil compaction stress is something which a plant can experience throughout their life cycle. So, for example, I'll see if I can get a pointer here. 1 second. Go. Pointer. Visible. There we go. So it's not, can you see my cursor? So here we can see a seedling growing. And for example, if you have a surface crust, which is a compacted soil property, you can obviously, that's going to affect soil germination. Compaction stress is also going to happen when roots reach layers, for example, plough pans, where there hasn't been tillage, and this is a barrier to further growth into the subsoils. So there are many aspects of soil compaction which a plant will experience during its life cycle. So here's some examples of some soils. So this is a 1 meter long soil column taken as part of the Developing Future Wheat program. And you can see what we've done is we've CT scanned them. These are 3 columns you can see here, 1 meter long length. And you can see how the top parts of the soil columns are more the greater air spaces. For example, you can see in the black, the gray is the soil content, and you can see it becomes progressively more solid, fewer air spaces, the deeper it goes. And this reflects the plough pan down to 30 centimeters. And then later we have the subsoils, which are a lot more compressed. Now, the compacted subsoils have a big effect on wheat root growth. And this is what this was looking at. And this is illustrated here when you measure root density, length densities, for example, which are at the different positions along the soil columns. You can see that there's a massive drop off below the tilled zone, which is about 20 to 30 centimeters. You can see that the levels of rooting really struggle to go deeper. Interestingly, and this is work that was originally flagged by Kierkegaard and Philip White, but also before that by John Paciori. Roots in the subsoils tend to prefer to occupy biopores, and these are some CT scans of soils. You can see here the roots, which you can visualize through this CT scan. You can see within the biopore the roots. Many roots are actually growing through here because they prefer to penetrate the deeper subsoils using this route to do so. It's estimated, Philip's estimated, between 60 and 90% of roots tend to occupy this means to do so. Now, compaction isn't just about subsoils. Compaction can also happen in upper soils, and this is where management is a really important factor to try and ameliorate this. But for example, heavy harvesters can have a really negative effect in terms of this and impact later yields. So what's the impact of compaction on root growth? And this is where we came into the equation. We started to study this process. We were looking at how roots adapted to compaction stress. And on the left-hand side, you can see my cursor, we have a CT scan through a column, and we've highlighted the root of a rice plant in yellow, and you can see it through this non-compacted soil. It's about 1.1 bulk density. It's nice fluffy soil. You can see the soil particles, the air spaces. You can see the root is able to penetrate quite happily. In compacted soil, and what we've got here is a 1 centimeter upper soil This is where the seedling germinates, and then later on we have the compacted soil layers, which are up to 1.6 bulk density. You can see the root is clearly struggling. And if we excavate these roots and have a look at how they look, you can see quite clearly the roots are getting shorter as the level of compaction increases from 1.1, 1.3, and 1.6. So we see its reduction in root length, and that's obviously going to have a big impact on root surface area, which is going to mean there's less nutrients and less water taken up, which is obviously going to explain why there's less yield when roots are exposed to compaction. One other characteristic of compaction is that the roots swell. And this is some very nice initial work by Bipin Pandey, who's now a BBSRC Discovery Fellow in the team. And you can see Bipin excavated roots, and then he did cross sections and visualized them using this in confocal microscopy. And you can see if we take the roots from the meristematic zone, you can see the roots are about the same radius, irrespective of their compaction level. But as you go into the elongation zone and differentiation zones of roots, you can see there's this massive expansion. And this is largely through cortical cell expansion. This takes place. And when Bipin was showing me these images, it just reminded me of of work that I'd seen and also performed as a researcher, where the effects of a hormone called ethylene. So as you can see, just like compaction, ethylene triggers, when you expose roots to ethylene, you see this expansion of the radius of the root, shortening of the root, and expansion of cortical cells, as I described for compaction. So this gave us the first clue that maybe ethylene was involved in triggering this adaptive root response. So the obvious experiment for me as a molecular biologist and Bipin was to say, well, what happens to soil compaction stress responses in roots if they're no longer sensitive to ethylene? So we were fortunate to be able to acquire some ethylene mutants. So here we have the ethylene signaling pathway in plants. It's well established across higher plants. You can see ethylene binds to the receptor, and this results in the signaling cascade through to stabilization of these transcription factors, which then trigger ethylene responses. If we knock out AIN2, we remove this, we can create an entirely ethylene insensitive plant. And you can see, so if we take the ethylene insensitive mutant and grow it in non-compacted soil, it's happily growing just like its wild type equivalent. Remarkably, if we expose these different genotypes to compaction, we see a very different response. In contrast to wild type, which you can see is struggling to penetrate the compacted soil, in the case of the ethylene insensitive mutant, you can see it's completely oblivious to this compaction stress. So this is actually a really surprising result, because a huge literature has concluded that part of the reason that roots will stop growing is because of the physical force required to penetrate compacted soil, also the difficulty to extract water under these circumstances. So there are lots of biophysical reasons why roots might struggle to penetrate compacted soils. But clearly, in the ethylene mutant, it's blind to this, it's oblivious to this. So clearly the physical limitations are not, in fact, the reason why the roots stop growing, because clearly in the case of the ethylene mutant, this isn't the case. So we conclude from this result that roots are able to penetrate hard compacted soils, up to a point, I should say, but they normally elect not to. So essentially, roots normally stop before they really have to. This is in an ethylene dependent manner. So if you make the roots ethylene insensitive, they're able to carry on and grow through the compacted soil. So just to illustrate that, in fact, when the root is exposed to compaction stress, we trigger an ethylene response. Within the root itself, we've created rice ethylene reporter lines. So in this case, we've tagged with GFP, the ethylene transcription factor, which is beneath IN2. And you can see that when ethylene signaling pathway is activated, this protein becomes stabilized, and you can clearly see it during compaction. It's becoming stabilized, and interestingly, it's in this elongation zone in this rice root. And this is the very nice work which we did in collaboration with Guoqian Huang from Shanghai, Jiaotong University, when he was part of Dabing Zhang's laboratory. So the question is, how does ethylene signaling increase during a root compaction response? So it's possible that this ethylene response is somehow upregulated in response to mechanoperception. So, for example, if ethylene levels were dependent on a mechanoreceptor, which was sensitive to the hardness of the soil, that's one mechanism. So we wanted to understand how this ethylene response was triggered. So the first thing we did was actually measure levels of the ethylene precursor ACC in root tips which had been exposed to compaction stress. And the conclusion was the ethylene precursor ACC levels remained unaltered during compaction. So there's no significant difference in the levels of ACC during compaction. So this clearly wasn't the basis. The upregulation, for example, of ethylene synthesis wasn't the basis for this sensing response. So rather than this ethylene response being controlled by the plant itself, we asked the question, could the enhanced ethylene response actually be due to changes outside of the root itself? So this is a rather radical idea, and it basically came from long discussions with Sashimuni about the changes in the structure of soil during soil compaction. And it made me think about the way gases diffuse through soil, because when a soil is compacted, what happens is the interconnected pores spaces are reduced massively. And so this cartoon illustrates what happens. So roots are known to pump out ethylene into the rhizosphere. It's been well known since the 1980s or even before. But the role of this ethylene wasn't clear. And the mechanism we proposed was maybe this ethylene is acting as a sensor for compaction. So when the soil is nice and non-compacted, this ethylene will diffuse away. But in compacted soil, the ethylene can't diffuse away, and it actually accumulates around the root itself. And this is what triggers eventually an ethylene response and all these adaptive responses I've described. So we set about testing this idea. And first of all, we wanted to understand, well, what happens to soil under our conditions? So through using CT scanning, and this is Craig Sturrock, who hails from up in Scotland and has worked there. Many of you know him. So Craig did a very nice analysis of the soil matrices during compaction and non-compaction. And you can see that in compacted soil, the colors illustrate the interconnected pores spaces. And as you can see in non-compacted soil, there's a lot of interconnected pores. But in the compacted soil, there's very little. And this means that diffusion of gases is significantly reduced in compacted soil. So using these images, a French collaborator, Olivier Martin, who's a mathematician, modeled these using soil models. And he was able to see that there's an interesting cutoff. As you get around about 1.6 bulk density, there's a very large reduction in bulk effective diffusion rates of ethylene in soil. Now, obviously, these are imaging and mathematical based simulations, but what happens experimentally? Were they correct? So some colleagues in Holland, this is Rens and Sean, they set about testing whether compacted soil really did provide a barrier for gas diffusion of ethylene. And they developed a very simple experimental system where they took a gas chamber, which you injected ethylene into. Then you had a kind of soil samples which were placed within this neck, which then linked a second gas chamber here. And they measured levels of ethylene moving from the upper gas chamber to the lower gas chamber. And what they observed was really significant. If you actually have no soil in this interconnected chamber, you can see that the ethylene from the upper chamber you inject rapidly equilibrates with the concentration of ethylene in the lower chamber. You can see here within a matter of 100 minutes. If you put non-compacted soil within this interconnected chamber, you can see it does create a diffusion barrier, but eventually over time you actually do get the gas diffusing. Interestingly, if you have compacted soil within this chamber, over 100,000 hours, we were shocked about how effectively nil gas diffusion occurred between the upper and lower columns when we within this interconnected column there was compacted soil. So this really provided evidence that soil compaction really does reduce gas diffusion within soil. So this is a really kind of surprising result. I tell my students often that plants are not green animals. They've come up with very original ways to sense their environment. And what they're doing here is essentially using their changes in the external structure to monitor a key parameter, in this case, compaction. They're using a diffusible gas signal. And I guess what's critical to this idea is that it's not a mechanical stimulus, it's a change in gas diffusion. And so one way to mimic compaction stress is simply to cover the root tip with a non-gas diffusible matrices. We used high vacuum grease, and we covered the root tip, and we were really pleased and to see that there's a rapid increase in the levels of an ethylene reporter gene when we do this. So this demonstrates that soil compaction resistance as responses are triggered not through mechanical stimuli, but actually through changes in gas diffusion. So here we have a kind of cartoon for what we think is happening in soil. So during non-compacted soil, on our left, you can see normal gas diffusion. The ethylene is produced by the plant root is released, and then because they're interconnected pores, it's able to diffuse away. In compacted soil, we get eventually an accumulation of this ethylene. It's not able to diffuse away, and this triggers a number of adaptive responses. So I guess ecologically, you could imagine this. Ethylene's almost acting like a stop signal, which then triggers adaptive responses. Why do people want to do this? Well, essentially, if you imagine you're a plant that's growing in natural soils, if you're a root that's growing in compacted soil, this is probably a waste of time. And by having a stop signal like ethylene, this allows you to say, okay, well, this is just a waste of time. Let's put the resources the plant has into other soil, other roots, which may be better placed to acquire nutrients and other resources from the soil. And so this is a very kind of effective way within a heterogeneous soil environment. The problem is within crop plants, which are obviously growing within a more uniform soils, you'd imagine this is actually a real problem, because once it hits the plow pan, they're going to obviously encounter problems. And this may, if we can overcome this, this would be really, really excellent. And I'll talk about that later. So you may notice anatomically the changes which are taking place in response to ethylene. So the cartoon illustrates how the root becomes shorter and thicker. These are two of the major growth changes which take place. And this is due to reduced cell elongation, which is triggered by compaction, but also this cortical radial cell expansion, and that's the reason the roots become thicker. So to cut a long story short, for what's behind this, we've identified that ethylene itself doesn't trigger these changes. It actually uses other signals downstream, acting downstream of ethylene, to do so. So in a recent paper in PNAS, we described how the ethylene signaling pathway actually, in response to compaction stress, this results in the stabilization of this ethylene transcription factor. And this directly binds to an auxin synthesis gene called YUC8, which upregulates the levels of auxin. And this auxin then is mobilized to the tip and back through the elongation zone through using auxin transport pathway to actually trigger the auxin-induced cell elongation inhibition. So this is the basis for the reduced root growth. In contrast, the actual swelling response appears to be mediated through a different hormone signal. So ethylene in this case triggers, we think indirectly, the upregulation of ABA synthesis genes, and that results in the accumulation of ABA within the stele of the plant, which then is mobilized out to the cortical tissues, and this triggers this swelling response. If you mutate these ABA synthesis genes, you can actually disrupt this radial swelling response. So here we see ethylene coordinating a number of different downstream signals to trigger growth inhibition, but also radial expansion. So just to summarize this part of the talk, soil compaction is obviously a major problem in agriculture, which can be addressed through changes in management practice. But obviously what we're hoping to do is to try and develop genetic solutions to this process. Our results reveal that root growth arrest during compaction is actually not due to physical limitation, at least in the levels of compaction that we're talking about in this talk today, up to 1.6 bulk density, but it's actually a choice. The plant's making a choice to stop, as I've illustrated earlier. And ethylene plays a critical role in this process. And this allows the release of the ethylene gas into the soil, enables plants to sense soil compaction indirectly. And reducing the sensitivity of plants to ethylene actually enables the roots to penetrate highly compacted soils up to about 1.6 bulk density. So this obviously opens up new opportunities to breed crops which have reduced sensitivity to compaction stress, and I'll talk about that at the very end. So I'd just like to underline that all this work, this kind of novel mechanism was very much a product of discussions between soil scientists, mathematicians, engineers, computer scientists, and plant biologists. We couldn't have come up with this without this dialogue taking place. So I'd like to move on now from talking about the mechanisms which underpin compaction. I'd like to talk to you about this new idea about root radar. And what do I actually mean by root radar? Well, I would argue that ethylene is actually functioning like a root radar signal. So what I'll describe is the definition for this idea. So we define that root radar signals function as these are signals which are released into the soil, and their levels change in response to soil stresses. And obviously ethylene is released, as an example, is released into the soil. Its levels change in response to soil stresses. I've talked about how changes in soil structure during compaction stress trigger the accumulation of this around the root. This is perceived directly, or it could be indirectly by plant roots, and I can elaborate that during questioning if you'd like. And then obviously what's critical is that there's a triggering of a root adaptive response. So what we propose, so ethylene clearly fits into this category. It's signaling back to the root when there's a soil stress, but we think that there are many other signals which may also function in a similar way. And some very interesting work that addresses this question: do other hormones function like root radar signals? Well, we know. A lovely paper by Tim George and colleagues described recently how there are almost 20 or more hormones which are detectable, which are released from the roots into the rhizosphere and the bulk soil, and can accumulate some distance away from the root itself. And this begs the question, what are the functions of these signals? Could they be playing roles to sense soil stresses? Could they be communicating back to the plant or to the microbiome? And so this really, I think, raises some fascinating questions. We know, for example, that some signals which are released into the soil do function like root radar signals. Strigolactones, for example, in a very nice paper produced by Tom Bennett's lab, they've described how release of this from seedlings has a very important role in the way that plants can sense the soil volume they're growing into and to adapt their growth. So here we have an example of a soluble signal, strigolactone, being released from the plant, and then the plant sensing and its neighbors the density, the volume of soil that they have, and adapting their root growth and shoot growth accordingly to the limitations presented. So I guess one of the questions that we would like to address is how far do these signals actually travel in soil, or are they required to travel in soil? So you can imagine a root that's in a soil volume. How far does it go? Does it go beyond? Does it need to go beyond the rhizosphere, such as section 1, or does it go into the further distance away? Do they create gradients? Do these gradients change in response to soil stress? These are questions we'd like to really address. We know that the rhizosphere is a highly dynamic zone which can change its properties as a plant gets older, and this may affect the movement of signals into the bulk soil. And how does this affect root radar signaling? And obviously we need to be able to measure these, and obviously we'd like to develop capabilities to do this. And here's one example of where we're imaging positioning of these sampling probes to be able to do collect samples and do mass spec analysis on them. So what I'd like to simply leave here is that ethylene is likely to be the first of many root radar signals. We have evidence for at least 2 to 3 more of these signals, and we really think we're scratching the surface of this new class of signal. I guess this is almost something which is a mindset of a plant biologist. This is an example of where we think that plant signals, their functions stop at the surface of the plant, within the root itself. And I think that measurements of that Tim and others have done on signals in the soil beautifully illustrate that plant signals don't stop at the epidermis; they go beyond. Into the rhizosphere, and then that begs the question: what on earth are they doing there? Do they have a function? And I think this is going to be a really exciting area of biology to characterize, to understand their functional importance in this space. So I'd like to shift now and talk about how we can exploit this mechanistic insights into how plants sense soil stress to improve resilience in crops. And obviously, let's go back to the ethylene example. Can we create soil compaction stress resistant crops if the roots are no longer sensitive to ethylene? Now, a lot of the work we've done so far has been done in models like Arabidopsis and rice, but we've demonstrated in rice that indeed, if we alter ethylene sensitivity, we can confer compaction resistance. Here we have another paper, and this was done in collaboration with Glenbain Goch and Ken Loades, and James Hutton, and led by Jonathan Lynch. And you can see in this case, rather than use mutants, and we used a pharmacological intervention. So this was done by a talented student based between Nottingham and Penn State, and James Hutton. called Doreen Van Hees, and she did some lovely work in field trials in Arizona, where she showed that if she actually treated plant roots with an ethylene inhibitor called 1-MCP, she could generate compaction resistance. So this really provided very nice evidence that when roots, and this is in the case of maize, so now we show that interfering with the ethylene response in maize, as well as rice and Arabidopsis, we're really seeing a pattern here, which illustrates the possibilities that we could exploit this mechanism in major cereals, at least, to engineer compaction resistance. So we've taken this one step further, and we said, okay, well, do we have enough natural variation in ethylene sensitivity of roots? And so we've recently, with the help of the guys in John Innes, Simon Griffiths and colleagues, we've screened the entire Watkins collection, and here's just a subset of that. And you can see if we've looked at over 800 different Watkins varieties, and we've looked at their ethylene sensitive root growth, and you can see there's a vast range of sensitivities within the land races. So you can have some lines which are very insensitive and other lines which are very sensitive. And we were intrigued by this because this now provides us with the ability to ask this question: does root ethylene sensitivity provide a good proxy for soil compaction sensitivity in cereal crops? And the upshot, the answer is yes. So if we take ethylene insensitive lines and we expose them to compaction stress, you can see that there's, in this case, there's negligible differences. But if we take lines which are sensitive to ethylene, we can also see that they are sensitive to compaction stress. So this appears to be a really quick and easy screen for compaction resistance in cereals. So this begs the question: what happens in elite lines, not just land races? How do roots of modern elite lines differ in their ethylene sensitivity compared to the land races? And we were really intrigued to see that if we take the lines from the UK recommended list, these are spring wheat lines, you could see that the majority of them are within the upper quartile of all the ethylene sensitivity which we saw in the land races. So in effect, breeders are indirectly selecting, it appears, for less ethylene sensitive varieties based on presumably their improved field performance, as they're looking for lines which perform across entire fields. They want uniformity, and we know that there's a huge amount of variation in compaction levels across fields, and you're effectively selecting here indirectly, probably for reduced ethylene sensitivity. So we're currently ground truthing whether we can take wheat lines which differ in their ethylene sensitivity and improve compaction resistance. So these are just some pictures taken a couple of weeks ago by colleagues in ADAS of our field trials, which are happening in north of Nottinghamshire. And this is a BBSRC grant, which we've just started, called Breakthrough, which is between ADAS, Rothamsted, Scottish SRUC, and several universities, Lancaster and Nottingham. So I guess one question which puzzled me is, why do we see such wide variation in root ethylene sensitivity? This was really quite striking, the variation. And some work that was done by two very talented members of our team, Lucia Nevis-Gannon, who is a PhD student joint with Simit, and Matthew Reynolds, and Rick Fusey, who's the postdoc on the Breakthrough grant. They asked the question, well, could this phenotypic variation be somehow related to the origin, the site of origin of the wheat lines we're looking at, and particularly the soil properties? And so they've done a very interesting pilot study where they've taken phenotypic data and then related that to the sites of origin of these different Watkins collections. So they've geolocated these for 150 of these genotypes, and then they've related this to the World Soil Database. And they've analyzed to see is there a correlation between ethylene sensitivity and a soil property. And textures has come up as a really intriguing feature. You can imagine that texture has a profound effect on gas diffusion. Indeed, in this very simplified cartoon, you can see here that particle size can have a very marked effect on the ability of gases like ethylene to diffuse. And what we've done is we've taken the USDA texture classes charts, and we've used these to look at the types of soils in the topsoils that from this analysis. And what we were surprised and intrigued to see that if you have lines which were isolated originally, sampled from soils which were a finer texture, they were more ethylene inhibited versus ones which came from coarser sand horizons. So it appears that wheat land races adapted to finer textured topsoils are more sensitive to ethylene compared to the coarser textures. And so we were obviously we would like to get more lines to have a look at the strength of this correlation, but this looks really quite promising that the soil texture could be a very important diagnostic for these differences that we're actually seeing. So general conclusions, I hope I've illustrated to you that hormones play key roles translating environmental signals into adaptive growth responses, and I've given you some examples about ethylene and ABA and auxin. I've shown you how plant roots employ the hormones in ingenious ways to sense stresses and how they use this gaseous property of ethylene to sense soil compaction. I talked a little bit about root radar and how we really think it's a new way of thinking about root and soil interactions, and how it could provide the key to sensing a range of different soil stresses, and how understanding these mechanisms really could provide new opportunities to create stress resilient crops. And so we're fascinated to study this in other cereal crops, and Tim George visited last week, and we're really interested in working with him to understand, for example, how barley may perform and whether there's this strong relationship between ethylene sensitivity and compaction resistance in barley, as well as we've seen in wheat and maize and rice. So just to thank many people, the ethylene gas diffusion mechanisms. If you're interested, there are really two papers which underpin this. This is the one we had in Science in 2021, and then a more recent PNAS paper talking about the downstream signals. And there are many people to thank for this, but I'd just like, for the interest of time, to flag up one in particular, Bipin, who really has been the driving force behind this. But I'd also like to thank Sasha Mooney, whose insights and had been instrumental in terms of telling us about how soil and gas diffusion properties are inextricably linked. The Breakthrough Grant is very much a collaboration between Nottingham, Lancaster, SRUC, Rothamsted and ADAS. And thank you to all those colleagues who have been actively involved in this. And the Watkins work, I'd like to thank in particular Lucia and colleagues at CIMMIT, but also we'd like to thank Simon Griffith, Lutzi and Shifeng for their generous help. And finally, the root radar idea. We currently have that idea is actually out to review at the moment for a Lola award, and so we're currently on the advanced stage of that, and we're hoping that we get a positive decision, because we think it's a really fascinating conceptual advance in root soil interactions. The idea for root radar, I'd like to acknowledge Tom Bennett. I think he captured the idea, and I'd like to acknowledge that. But most of all, I'd like to thank Bipin, who really has been a huge driving force for all this compaction work. And I'd like to congratulate him recently getting tenure and a Nottingham Research Fellowship. So hopefully he'll continue for many years at Nottingham and continue this rich seam of research. Thank you very much.
Thanks for that, Malcolm. That was really entertaining. I think everyone would agree it was quite enjoyable. It was nice to see a mix of different disciplines as well. So you got the physics and the maths involved in that, and the modelling. That's the way it should be, and doesn't happen enough, as far as I'm concerned.
Oh, as I said, we couldn't have done it without, those conversations with Sasha were absolutely vital to think about, the answer laid outside the root, and that's, I think, one of the big problems in plant science at the moment is that we don't realize that, soils play such a vital role, and many of the answers are actually out there, but it requires us to engage with soil science. And I think Hutton is one of the few places which where that takes place, that dialogue takes place.
Yeah. Cool. Okay, so we've got some questions coming in. So Raul Huertas, he's asking, when it comes to investigating roots, so do you think that root radar signaling concept might prevail to other signals like nutrients, water?
That's a really interesting question, and we think that's the case. Without giving anything away, we've identified some really interesting signals which are upregulated massively in response to nutrient stress, and we think they operate as a root radar signal. And we also think that signals like ABA need to be rethought. Tim very nicely showed in that paper I flagged that ABA it doesn't stop at the root epidermis, it goes out into the rhizosphere. It could well be having impacts on reprogramming the microbiome or signaling back to the root about water stress. And I think that this is a really fascinating area that we can really need to start to think about, go beyond the root and think about, these are inter-kingdom signals potentially.
Yeah. Cool. Raul, I think you've got another question. Do you want to ask your follow-up?
Yes. So basically, when we're growing plants, we only consider the soil the only properties the plant need to grow well. That's the only thing that we care. So to what extent is possible that we mimic the physical property and the soil structure when we are phenotyping potted plants?
Oh, sure. I hope I've given you encouragement to think about using lots of different textures. We've literally scratched the surface. We tend to use extremes, soil extremes, so sandy loams and clay loams, but there are many different textural types which arguably you need to think about. There was a beautiful paper by Andrea Carminati recently where he looked at the root adaptive responses to water stress in 9 different soil types, and there were major differences because all of them have got very different water release properties. And as plant scientists, well, I'm a biochemist by training, so it's only talking to soil scientists that I've started to really understand the differences in the properties of these soils are massive and they have huge impacts. So you're absolutely right. We need to when we do these experiments, we need to think about using lots of different types of soils. Thank you.
Cool. Look.
Great talk. Still with a plant-centric view, was there a genetic signature associated with the ethylene sensitivity in the Watkin lines? I mean, ethylene receptors maybe, or
yeah, that's a great question, and in fact, yes, there was, this is where we really were helped massively by the sequencing of the Watkins, the WATSEQ initiative, which is Shifeng Cheng has really driven with Simon and colleagues, and we were able to access. They've sequenced 900 of those Watkins collection, and we were able to then, through their GWAS analysis, identify a region which actually controlled this. And we've donated this information to the Breeders Toolkit as part of the wheat Yeah, and it's now being used. This is way in advance of publication. And we think it's really important that they, breeders have access to this and try and bring these alleles into the germplasm. I mean, probably the alarm bells were going for many of you thinking about making an ethylene sensitive plant, because obviously it's so important for disease resistance, for mycorrhiza interactions. So I wasn't surprised in a way when we found the genes that we landed on. They appear to be downstream of the primary signaling pathway. So they're not the primary signaling machinery. So that remains intact and operating. And we think that there's a secondary pathways which go off to different functions. So we think that we're getting allelic variation actually in components which are downstream of the primary signaling pathway. So the disease resistance is still intact. The mycorrhiza interactions are intact. This appears to be affecting just the responses to, as the cells are expanding in the elongation zone. And I think these are the alleles that we want. We don't want to disable the ethylene disease resistance. That would be ludicrous. So this is, I think, the power of genetic variation coming to the fore here. We're selecting alleles that we want rather than go after the primary signaling machine, which would be madness. Yeah.
I think as a geneticist, that's the answer you wanted, Luke, I think.
Yeah.
So there's an interesting question here from Ruud Korver. He's saying, do you think the positive effect of intercropping might be due to crops reading each other's radar?
That's a great question, and absolutely. Again, we anthropomorphize plants, don't we? And we think about them acting as individuals, but they operate at the field scale, at the population scale. And there's lots of evidence that many signals, for example, the strigolactones, are acting for interplant communication. So I alluded to this Lola grant that we've got under review at the moment. And we are really, when we're looking at the root radar signals, we're not just looking at individual plant responses, we're also looking at interplant responses, because I absolutely, they will play a role at the population scale too.
Okay, cool. I'll ask you maybe a daft laddie question, because I always like to ask those, they're the most difficult. On these ethylene insensitive lines in the root area, are they paying a penalty above ground because they're ploughing on regardless below ground?
That's an interesting question. We were really, so this would be some great work by Ian Dodd, for example, showing that when you expose roots to soil compaction, signals move from the root to the shoot, which actually inhibit growth. So he's measured ACC and ABA, for example, that it increases. So we were very intrigued to see what happens to the shoot responses in these mutants in response to compaction stress. And what we observed is that they kept on growing. They were far less growth inhibition in the ethylene sensitive lines than the actual wild type. So this growth penalty, which you saw in response to compaction in wild type, we saw a much less reduced. Well, there was some reduction, but it was much less. So it suggests that not just getting reduced ethylene sensitivity in the root, but also in the shoot may actually be a benefit.
Okay, cool. Because I was thinking of flipping it one way, that if you did get, and you use the ethylene sensitivity in things where people are producing things through hydroponics, because you don't want massive amounts of roots, but you want massive amounts above it. And that'd be a controlling system to flip. I don't know.
That's an interesting idea.
The bit you were talking about in soils as well was interesting, and I think it's and I'd imagine most of the plant people here would agree that our understanding of soils is not great because it's not our area. But I remember reading stuff previously. I mean, a dry soil can be sitting at an alkaline pH, whereas a wet soil is probably going to be kind of acidic. So your ethylene is going to be pretty sensitive to alkaline. I know that as a chemist. So if you're looking at climate change and you're flipping between dry and wet, you're going to get floods and bursts or removals of ethylene, I kind of guess, from a purely hypothetical basis.
No, that's an interesting point. And we know that ethylene is also an important signature, for example, in hypoxia. So you can easily imagine, when the pores, all the soil pores are flooded, you're going to get even less gas diffusion. And one of the questions we got when we first submitted the work to science was, well, are you just triggering a hypoxic response? And so we took hypoxic markers and we showed that compaction doesn't trigger a hypoxic gene response. We use loads of markers and they're quite different. But you're right. It could be that the chemical properties of soils may interfere with this signaling mechanism. So without letting any secrets away, we've identified additional volatile signals which control compaction responses. So ethylene isn't the only volatile signal which the plant uses.
It'd be interesting to see with soils that have got biochar in them, which are hyperabsorbers, how they behave, or how the plants behave in that way.
That's a great idea, and maybe that's where they really provide benefits. You could imagine that for bacteria do the same thing. One of their active effects, again, this is Ian and other colleagues have done some lovely work showing ACC deaminase is a very important enzyme based by the soil bacteria. It breaks down the precursor to it. Bacteria producing these kind of enzymes makes a lot of sense, because you want to remove these stop signals. The bacteria want the plants to invest carbon into the soil, and so they're taking the brakes off, aren't they? And
absolutely. I find it fascinating that one of the world's simplest gases is still being argued about with regard to what it's doing. I mean, it doesn't get much simpler than ethylene, does it?
No, that's true. That's true.
Okay, I think we'll let you off the hook now, Malcolm. That's been a fascinating talk, and I think if everyone can just show their appreciation in the normal fashion, thanks very much.
Thank you.
And I'm sure we'll meet up again, and we look forward to hosting you in the flesh at some point.
Thanks very much. Thanks, everybody.
Thanks very much,
Thanks for the
Malcolm.
great questions. All
Thanks,
right,
everyone.
take care, Derek. Bye-bye.
Bye.